Hello everyone, welcome to my YouTube channel aksharaaalaya. Today we are going to discuss about the interesting topic called cell. We are discussing the introduction to cell. This topic is selected for this session only because cell is a unit of life. Without cell, we cannot discuss the topics of biology. As biology is study of living organism or living system, all living system contains a building block called cell. There is a disclaimer before going to this topic. This session is purely based on how the cell was found and the story behind the invention of cell. As I believe, knowledge is not only about filling your brain with terminologies and the definitions. The real knowledge is bringing out the curiosity in you to learn new concepts. How can I bring the curiosity of learning biology or learning cell is by explaining the story of invention, story of research behind every topic. If you know the background of that topic, you can easily go through the subject and you can understand the subject more than just reading the subject or memorizing the terminologies. You can really understand why, what and how. So going to introduction to cell. As we know, cell is in every living being. There is no muscle, no nerve, no organ without a cell. Cell will form tissues, organs, organ system and organism. And also some organisms are unicellular. A single cell will act as whole organism. You may have seen the eggs of hens, ostriches, which are single cell formed from the fertilization. And many cells in our body, blood and other parts are unable to see in naked eyes. We need a special what instrument called microscope. Without microscope, we cannot see the cells. As scientists see the galaxies and stars very far from here in the telescope, they are not seeing the present. They are seeing the light of past. The stars may have emitted the light from million years ago. That light have been received now and they are saving it now. So scientists are seeing in the telescope that is the past of the universe. In the microscope we are visualizing the minute details of the whole different world. That is, with the help of microscopy we are studying the cell, which is the unit of life. As to understand the whole universe, it is very difficult to know. But we are understanding the part of universe to know the characteristics, how this world works. So, how the cells were formed or how they found by the scientist. Till the 16th century, there was no microscope found, or in the till the mid centuries of 16th, there were no microscopes and there was no much idea about cells. But Robert Hooke, who was a curator in Royal Society of London, wanted to know how the cork of bottle will not allow the contents of bottle or air of bottle to get out. What is the nature of cork that stops the matter from flowing out? So he took a piece of cork and observed under a microscope. The microscope was invented very recently and he used that to analyze the structure of cork. He saw some block-like or hollow structure called cells. How he named this structure as cells, it is not because he didn't know they were living and they were unit of life. He just saw those structures. Those structures was similar to the monastery where the monks were residing at that time. Monks were meditating, praying the God or Almighty in those cells, means room-like structures. He have noted his notes. I will repeat the sentence for you. He have noted in his writings that I took a good clear piece of cork and with a pen knife sharp end as a razor, I cut a piece of it and then examining it with microscope, me thought I could perceive it to appear little porous, much like a honeycomb. He sees it as a honeycomb-like structures which are little porous in nature. He named him as cells, following to cells resided by the monks at that time. So cells was not intentionalized. This term was not intention to describe the living thing, but it came from the idea of the monk residing in monasteries. Parallelly, there was another scientist called Antonie Von Leeuwenhoek. Antonie Von Leeuwenhoek was making his living by selling clothes and making buttons for the new clothes and all. He was grinding glasses and making lenses for as his part-time interest. Let's say it as a hobby. He was making lenses and designing some microscope by himself by keeping the lenses one upon another to making the light to bend and give an enlarged image of small object. He was designing microscopes and he was finding the world of microorganisms. He didn't know at that time they were microorganisms. He took a drop of water from pond and observed under his microscope, which was not much efficient or effective because of less technology, but he knew there was something beyond our eyes and there is a whole new world to perceive or to know. So when he have seen the drop of water from pond, he have seen small objects which were living. He called them animalcules. He called them animalcules because molecules, animalcules, as it is a small part of all smaller version of animals, he said them as animalcules, and then he also discovered various shapes of bacteria at that time. Shapes of bacteria at that time. And he also wrote the letters to Royal Society of London. Royal Society of London, as we discussed, where the Robert Hooke was studying, or he was a curator. Robert Hooke was curator at that time in Royal Society of London. He sent the letters describing what he had found under the microscope. Over many years he sent letters after letters to Royal Society of London. They have had a doubt that these findings did not have any basis. So what they did, Royal Society of London appointed Robert Hooke to cross-check the information whether these findings are true or they are just based on nothing. So when Robert Hooke examined his readings, when he himself re-experimented what Antonie Von Leeuwenhoek did, he found that yes, there is a world of microorganisms and there are cells which are unable to see in our eyes. He contemplated it with his findings. His finding was the cells in the cork. And now Antonie Von Leeuwenhoek, as also said as father of microscopy, found new world of cells and their studies. But till the time of 1830s, there was no much relevant studies on the cell and its structure. But by the time of 1838, the German botanist Matthias Schleiden explained the structure of plant cells. By the time of 1838, he said that plant cells are covered by cell walls. There is a structure called cell wall covering the plant cells, and also he found that embryo was single celled. Embryo, which is a fertilized product of pollen and egg, was single celled. By this time, Matthias Schleiden said that plants and its other parts are made up of cells. But the study did not only stop to the plant cells. In 1839, German zoologist Theodor Schwann found that animal cells also have a membrane-like structure called plasma membrane. Theodor Schwann studied about the plasma membrane structure, and also he, that is Theodor Schwann and Matthias Schleiden, proposed the tenets of cell theory. Explained that they knew one thing, that is, all organisms were made up of cells. So first tenet was all organisms are made up of cells. And another tenet was cell was a unit of life. Because as it has an individual form and individual boundary, cell was called as cell is a unit of life. But they did not know how these cells were formed. They had a basic idea that cells were made of some non-cellular materials like biomolecules to say. But they did not know how the cells were formed and how they divided. So till the 1855, in 1855, Rudolf Virchow said that he was a German pathologist. He studied about pathogens. Pathogens are those organisms which cause diseases. Rudolf Virchow, after denying of the theory of spontaneous generation, he found out that cells are formed from pre-existing cells. So this was a third tenet of cell theory. That is, cells are formed from pre-existing cells. There is no spontaneous generation. It does not create itself from nothingness. It has some form, that is a cell will divide to form another cell. That is how tissue will form. They will form organ system and organisms. So In this session, we have gone into details of how the cells were formed. We have now know the story behind how the microscopes were designed and how Robert Hooke, Antonie Von Leeuwenhoek found the structures of cell, and there were further studies to build a cell theory by Matthias Kleeden and Theodor Schwann, Rudolf Virchow, who gave the cell theory. That is what we have discussed in this segment. I will give a link of the printed copy of letters sent by Antonie Von Leeuwenhoek for your reference, and also I will provide an image to see which type of microscope did Antonie Von Leeuwenhoek use to see the animalcules, as he didn't know the term microorganisms, animalcules at that time. So hope this session of introduction to cell helped you in enriching your knowledge. Next session will be on the cell components. Before that, we will study the properties of cell, how cell acts in a living system, how it contributes to living system. So hope this session was helpful. Please like, share and subscribe this channel, and please share this knowledge who are in need. Thank you.